All right. Well, thank you so much. It's really a pleasure to be here and also to kind of go back to face-to-face conferences where we can talk a lot. And today I'll kind of spend about half of my lecture a little bit more of a didactic background on the field. I recognize that many of you come from diverse areas, industry and academia and otherwise. And then half to talk about a new study which involves a technology that we developed and its application to study early stage breast cancer, TCIS. These are my disclosures. The only one that's relevant is this Resolve Biosciences company that I work with. I'll talk a little bit about some Resolve Biosciences data as well, which is a new spatial technology. As I mentioned, I'll give a bit of background on the field, talk about some computational methods that we've developed in order to analyze these large-scale data sets, and then one research project where we applied them to early-stage breast cancer. I like to start with an analogy, and that's one of a soup. As it gets a little bit cooler here in Houston, start to eat a lot of soup, especially minestrone is one of my favorites. SingleCell Genomics, in a sense, is different from bulk sequencing because with bulk sequencing, you take a tumor like this one here. This is a renal carcinoma. You actually put it in a blender and turn on that blender, and you mince it, and you extract out DNA and RNA. But it's very hard to tell where that DNA and RNA is coming from. There's many different cell types in a tumor. And so in the end, you have this mixture of components. There are some methods that you can deconvolute the data with a little bit to determine what's in there, but it's still quite hard to know the exact numbers of different ingredients. And with single-cell genomics, we can know exactly that each one of these ingredients can be thought of as a cell type. You know exactly how many beans there were, how many pieces of corn, to very kind of granular quantitative information. And so bulk sequencing so far has really been studied mostly to study differences from patient to patient. You may have heard of projects like TCGA, the Cancer Genome Atlas. This has looked at many, many different cancer types and just sequence one patient at a time and then look for common mutations across the cancers. And single-cell genomics, it usually used in a different way to look within one single tumor. You can look at lots of tumors, of course, but it's to resolve the diversity that occurs, the cell-to-cell differences that are within that tumor. And a tumor is often described as an ecosystem. There are many diverse cell types that can be present. There are the tumor cells, and so they can have genetic differences. We call those subclones. And there are also lots of different cells in the microenvironment, and these can be immune cells, connective tissue like fibroblasts, vascular cells that transport blood, lymphatic cells that take away waste, adipocytes, and other cell types. And so single-cell genomics, the idea is really to get at this diversity within the tumor. And as Vicki mentioned, one of the ways I got my faculty job was this early study that, where I developed a single-cell DNA sequencing method. This was done when I was a graduate student at Cold Spring Harbor Labs. Early on, it was hard to just even think about how do you actually isolate a cell from a tumor or a system. There were techniques like mouth pipetting, but they were quite tedious. Eventually, we arrived on FACS sorting, which could sort individual cells into wells and was fairly high throughput. It was used for subcloning at the time. Then we had to amplify the DNA or RNA. There's only about 6 picograms of DNA present. And so played around with lots of different chemistries that are called whole genome amplification methods, and finally arrived at one called DOP-PCR that can amplify DNA for copy number profiling. And then early on, I was mostly using microarrays to look at genetic differences in profile, but they never worked really well. And when the first next-generation sequencing technologies were developed, I started to use some of those. And actually, the early systems we used were mostly 454s. And we eventually switched over to one of the Illumina systems. But the nice thing about sequencers is they sequence all the fragments. So microarrays, they target specific regions of the genome, and if you don't amplify those regions, they fail to hybridize. But sequencing will sequence everything that's present. And so, and then eventually switched to kind of the Illumina systems and sequenced one cell per lane at the time, so it was quite expensive. But luckily, I had a great mentor who, after he saw the techniques were working, ended up kind of funding some of this project. We were able to then measure copy number profiles in individual cells. And the surprising result from this was that they were actually quite similar from cell to cell. These complex karyotypes, genotypes, within the tumor represented clonal expansions, as you can see over here. And so that was an early method to do single-cell DNA sequencing. After that, my lab has continued to develop methods to do single-cell DNA sequencing. We've developed methods to do whole genome sequencing or exome sequencing of the coding regions of the genome. Methods to do targeted sequencing of just cancer genes, which allows you to increase the throughput a lot. And more recently, we do single molecule single-cell DNA sequencing, and that improves the data quality quite a lot because you can actually determine which molecule is originally amplified from a single-cell genome. A colleague of mine, Fuqiu Tang, actually working even before me, but he was working in a very different field in embryology. He was working in Cambridge at the time. He developed some of the first single-cell RNA sequencing methods, and this took advantage of some early chemistry that James Eberwein developed. He's at UPenn, actually, but it involves oligo-DT priming of RNA molecules and then amplifying. And he, again, was using kind of an early sequencing technology, so this was the solid platform, which some of you may still be familiar with, but demonstrated that it was possible. And then over the years, single-cell RNA methods have really undergone a lot of technological innovations and development. Fuqiu's original method could only sequence about one cell at a time. He was mostly mouth pipetting the cells. Some of these methods were adapted for FACS plates, and that made them a little bit higher throughput. Stephen Quake and others developed kind of microfluidic platforms. Out of this spun a company called Fluidigm that was used for a lot of early single-cell RNA sequencing. Automation increased throughput a little bit, and then really the development of micro droplets or nano droplets that increased the throughput to tens of thousands of cells and was commercialized. Nanowells are another way to do this in high throughput. And then some new methods called combinatorial indexing can actually allow you to profile about 1 million cells in just one experiment. So it's kind of an interesting story about how these microdroplet platforms were developed. It was actually two groups, both working at Harvard, and two graduate students, Evan over here and Alan over here. They were working on kind of very similar approach in order to create microdroplets of oil and beads and cells that go in together and then amplify the RNA in a very high throughput way. They're working independently in two labs. They were not aware of each other's projects until there was a lab meeting and someone from the other lab recognized that it sounded like a very similar project. And then in a very collegial way, they got together, Mark Kirshner and Steve McCarroll, and they decided to co-publish their methods together in Cell back to back. And two methods were called DropSeq and InDrop. They're quite similar. One uses a solid surface bead with oligos on it. The other one uses this hydrogel bead. And this was commercialized then into 10x Genomics later on, which is the platform a lot of you might be familiar with. Another method that's quite exciting is called combinatorial indexing. Combinatorial indexing really is a method developed by Jay Shender's lab out of University of Washington. It involves doing chemistry in situ in the nucleus and then doing multiple rounds of fact sorting and barcoding to relabel cells. And using this through the combinatorial indexing, you can really scale up the number of cells that you can sequence. You can see here, if you do experiments with 384 by 384 wells in a plate, you get 100K cells. If you do 1536, you get up to 2 million cells. So that's really quite another massive increase in throughput for these techniques. Now, overall, the field of single-cell genomics has really undergone a lot of development over the last 10 years. Starting with some of the early papers, you can see they've just increased kind of exponentially. If we were to plot 2022, it's already way above that. It's really impacted lots of fields of biology and biomedicine. Cancer is just one of them. There's a lot of work in just normal development of species, neuroscience, microbiology, immunology, and still a lot of papers on computational tools and technologies. So what can you do with some of these methods? Single-cell DNA sequencing in the cancer world can be used to study intratumor heterogeneity, so look within a tumor at different populations and the combinations of mutations they have. You can then use this information to reconstruct evolutionary lineages and understand the order of mutations that occur throughout tumor progression, as shown in these lineages here. You can also look at clonal dynamics, so try to understand things like selection or neutral drift by looking at how populations change under different conditions, like say in primary tumors and metastatic ones. And then in the clinic, you can really use this information to look at the co-occurrence of different mutations within tumor cells. And this is important for things like targeted therapy, where you want to know if you targeted EGFR mutation, which cells are you actually targeting? Is it these over here, as shown as the case here, or what about these cells over here, which in this case you're not targeting? Now, the tumor cells, as I mentioned, are not evolving on their own, just in terms of their genetics. There's also the whole microenvironment, and that's also changing around the tumor. And some cells in the microenvironment will suppress tumor growth, and some might actually support tumor growth. And so this is an example of just the normal breast tissue. These are the lobules that produce milk. They transport out to these ducts, and you can see it's very differentiated tissue. As you get to premalignant disease, you see the malignant cells in the epithelial duct start to really expand. But you also see a lot of changes out here occurring in the connective tissue, the immune cells, and the stroma. And then as you get over here to invasive breast cancer, this is where kind of all hell breaks loose, and you see lots of dedifferentiation, many immune cells infiltrating, and many reactive changes that occur. And so single-cell RNA sequencing is a really great way to do an unbiased profiling of the microenvironment. In addition to getting the transcriptomes of the tumor cells, you also get immune cells like macrophages, T cells, NK cells. You get a lot of stromal cell types like fibroblasts, vascular endothelial cells, and others. And then just identifying cell types is pretty easy and straightforward, but you can also take them and then recluster the data and identify what we call cell states. And these are different biological functions within a cell type. Many of these are known for things like T cells, but when you think about fibroblasts or endothelial cells or myeloid cells, it's really not very well understood. And in the tumor cells themselves, you can also look at different properties like these phenotypes and see how they vary across the tumor cell populations. Now, a lot of early studies on single-cell genomics focused on looking at clonal evolution within the tumor. Some of ours are highlighted here in bold. And so these were studies looking mostly at the genetics and understanding the order mutations that occurred throughout tumor growth. A lot of the single-cell RNA studies, some of these coming from Avi Verjev's lab in Boston and others, they look more at the microenvironment, how that's impacting tumor development. And then another area is really looking at primary metastatic disease, understanding how tumor cells can leave the primary site and colonize different organs. Work by our group, also by David Ting, Dan Haber at MGH and others. And then two new areas I think are very exciting. One is pre-cancer, so understanding how really early lesions that you can detect by, for example, mammography, progress into the invasive cancers and predicting which ones are likely to progress and which ones will not. And then therapeutic resistance, where we can look often longitudinally at different samples over the course of therapy and understand responses to different drugs in patients. And there's a lot of potential to translate these into the clinic. They used to be very expensive, these methods, but the costs are coming down precipitously. And we're getting better and better methods that can work with frozen materials, FFP materials, et cetera. So there are opportunities in early detection. You can imagine any bodily fluid, you can look for premalignant cells. In your spit, that might be your lung cancer cells. In your urine, that might be bladder cancer cells. And then not just detect the cells, but actually profile and sequence them to see if they present a risk and harbor mutations. You can look at tissue and predict based on the tumor cells and the microenvironment which patients are likely to progress to invasive disease. You can do a lot of new drug target discovery. So because it's unbiased, you're measuring lots and lots of different receptors and other targets on tumor cells in the microenvironment. You can look at lineages of mutations and decide which populations to target. Do you go after the truncal mutations and try to eradicate all the tumor cells, or do you go after only the most malignant populations? And you can do a lot of monitoring in the blood over the course of therapy so that you can see how patients are responding. So to summarize the technologies here, we now have kind of a lot of these single modal technologies. We can do single-cell DNA sequencing, and there are commercial platforms like Mission Bio. There's also a lot of nanowell technologies, some that we developed called ACT, DLP. There's also some by 10x Genomics that they developed, but unfortunately this was actually pulled from the market due to some patent IP issues. For single-cell RNA sequencing, there's nanowell technologies. We actually use a lot of those. SeqWell is another one developed by Alex Shalick's lab at MIT. There are 10x Genomics, that's the most popular one for single-cell RNA, most widely used right now. And combinatorial indexing, these are just being commercialized right now with companies like Parse Biosciences. Single-cell epigenomics, there are good techniques to look at attack, so that's chromatin accessibility. Also single-cell DNA methylation and cut and tag, which allows you to look at chromatin based on antibodies that bind to the chromatin and then cut the DNA around it. And a new area is single-cell multiomics. So how can we measure from the same cell both the genotype and the phenotype? How to get DNA and RNA from the same cell, DNA and attack, or other information. And so these are just kind of being developed, and there's a lot of methods, early stage methods that can just analyze a few cells. This represents a very important new direction. So single-cell multimodal measurements, this is really where the field is heading. There have been a few methods to do DNA and RNA or RNA and epigenome. Actually, the first one of these was commercialized by 10x Genomics. It can do single-cell RNA and attack from the same cells, and it's very high throughput. And so a lot of new ones are kind of being commercialized and developed right now as well. And I think it's a very exciting time. The other area is spatial genomics, and so for these methods allow you to look both at the cells, their genotypes, their phenotypes, and then how they're spatially organized in tissue sections. And there are broadly kind of these two areas that are being developed for technologies. There are unbiased methods that allow you to look at the whole transcriptome, so you don't have to decide on the targets beforehand. Spatial transcriptomics, this is being developed by 10x Genomics. SlideSeq, that's from Evan's lab at Harvard, also being commercialized right now. And these don't give you single cell resolution, but they give you very small spots of cells that can be just a few cells up to 100 cells or so. And these technologies are being improved, and they can, pretty soon in the future, they'll be able to achieve almost near single-cell resolution. The other type of technology are in situ platforms that use kind of a single-cell RNA probes that are very multiplexed. These do give you single-cell resolution, but you have to decide beforehand what the targets are and what the probes are. These are companies like Merfish, Merscope, Seekfish, Resolve Biosciences, et cetera. NanoString, GeoMX as well. And so you're hybridizing lots and lots of probes. In the past, you could only look at a few targets at a time, but now you can look at 1,000 probes. Xiao Wei's lab with Merscope has demonstrated they can do 10,000 probes. So you're not really so limited anymore in kind of the target space as you used to be with these technologies. All right, so that's a little bit of background on the field, and now I'm going to switch over to some of the computational tools that we've developed, as well as some of the ones out there in the field. So just to put it in perspective, with single-cell genomics, the type of data that you get and the amount of data is really drastically increased over some of the traditional bulk methods. In a standard study, you would do one patient at a time, and you would measure, say, a genome or an exome and a transcriptome. And in a typical project, you might look at 100 patients or 1,000 patients, so 100 or 1,000 genomes. With a single-cell study, even in one experiment, you might profile up to 10,000 cells or even more. And in a project, which now is kind of approaching this kind of 50 to 100 patients range, you're really talking about 1 million transcriptomes and genomes. And if you just think of this in terms of one matrix, that has kind of rows and columns, you've really increased that quite a lot, and that makes it very hard for a lot of computational approaches to be very efficient. And so we really need a lot of kind of new analytical approaches in order to be able to analyze these large data sets. So a number of methods are being developed. On the single-cell DNA side, a lot of these come from our own laboratory, but there are also other people working on them. There really aren't that many methods. They generally look at copy number profiling or variant detection. Some methods by our group. Some have been developed kind of together with Louis Naclé here at Rice, also Ken Chen at MD Anderson, and others, Nico Bierenwinkel at ETH Zurich as well. For single-cell RNA, there's really been an explosion of computational methods. There's over 1,000 now, and these are just some of the categories. They range from everything from kind of data normalization to differential expression to everything else. And there was just a review paper that came out not too long ago saying, marking that there were about 1,000 tools that they had now tracked now. And you can see kind of, this number increasing here. Sometimes within a single category, like in trajectory analysis, there's over 100 tools and papers that kind of benchmark them. So there's quite an explosion of these tools right now. A little bit of a crowded space, but still there's quite a lot of needs for analyzing this data. So now let me just mention a few tools that we've developed in my own group. One of the first ones I wanted to mention is called CopyKit. That stands for copy number karyotyping from aneuploid tumors. And this was developed by Ruli Gao, former postdoc of mine. She's now assistant professor at Northwestern. And this uses a Bayesian Gaussian mixture model to try to infer DNA copy number information from single-cell RNA data. And this is a way that we can take single-cell RNA data sets. We kind of cluster the data together. We identify a single cluster as being diploid cells. We use the GMM, and then we try to identify different segments that occur. We decide whether to merge segments or not based on this MCMC. And after that, we identify events that are neutral copy number gains, or copy number losses. And that allows us to distinguish tumor cells from non-tumor cells. And so here's some applications. These are a bunch of different cancer types from collaborations at MD Anderson we worked with. This is triple-negative breast cancer. You can see the non-tumor cells here. They have diploid normal genomes. And the tumor cells up here that have different copy number amplifications in red and losses in blue. This is another case where we look just at the tumor cells now. And with these methods, in addition to distinguishing normal and tumor cells, we can also look for different subclones. In this case, there was a rare subclone over here labeled as B that had a number of additional amplifications, including this amplification that's clinically very relevant, which is the KRAS locus on chromosome 12p12.1. This tool is openly available. Lots of labs are using this now. It's quite useful for looking at single-cell RNA data. Another tool I want to mention is called CopyKit, and apologies for the similar name, but this one ends in kit. And this is a method for single-cell DNA sequencing analysis, not RNA. This was developed by former student of mine, Darlene Manussi, who's at Pfizer Oncology now. And this is kind of an end-to-end toolkit. You can think of it as SURAT for single-cell RNA analysis, but this is for single-cell DNA analysis. It starts with mapped reads. It does a lot of normalization of the data, segments the data, and then tries to find common breakpoints across all the single cells. It identifies diploid clusters, it filters out bad cells, and then it goes through this clustering optimization to identify the number of subclones that are present. It condenses the cells into individual clonal genotypes, and then performs different phylogenetic analyses. And this is just showing an application of a patient, MD Anderson, that had colon tumor and liver metastasis. We were able to reconstruct the different subclones that occurred during this metastatic process, and then really find kind of the links between the primary disease and the metastatic disease that's shown over here. And this is on bioRxiv. And then one more tool, this now gets into the spatial genomic analysis. This is a tool called CellTrek. This was developed by Runman Wei, a postdoc in my lab, together working with a graduate student, C1. And in this method, we combine single-cell RNA datasets and spatial transcriptomics datasets. As I mentioned, the problem with ST data is that it's large spots. It's not really single-cell resolution. But if you do the experiment from both single-cell RNA analysis and ST, you can then co-embed the data in high-dimensional space. You can train a model in order to try and assign different cells to different spots. And then it uses this multivariate random forest. And then is able to assign different cells based on this kind of distance graph to the spot that are most likely to occur. And that allows you to then get kind of single-cell mapping in this spatial transcriptomics data. And this is an example of a DCIS breast cancer. You can see different subclones that map to different ducts within the tissue. And this is an example of mapping different immune cell populations. And he showed that there were often these tertiary lymphoid structures that had congregations of both B cells and T cells that were neighboring the ducts in the DCIS lesions. Okay, and just to mention some of the computational challenges, I know there are a lot of computer scientists here in the audience, but challenges still remaining for single-cell DNA analysis include it's easy to over or under cluster the data. It's still very hard to optimize the number of clusters that represent subclones. Calculating DNA ploidy, so that's kind of the ground state copy number. So is your karyotype diploid or tetraploid or triploid? Still quite challenging with these data from read counts alone. Computing LOH and kind of A, B allele frequencies in single-cell data is also still quite challenging. And detecting kind of complex structural variants. Building phylogenetic lineages, still quite challenging, especially ones that integrate multiple sets of data, like copy number alterations and mutations and indels and other things. And then really still detecting somatic mutations is still quite challenging. Unlike copy number aberrations, these are at base pair resolution, and they suffer from a lot of technical issues, including allelic dropout. And then for single-cell RNA analysis, there's still a lot of challenges around clustering cell states. It's quite straightforward to identify cell types. Those are major clusters. But then to take that cluster, recluster it, and identify all different substates that exist is still quite challenging. Automating the identification of cell types is also challenging. It's very dependent on markers. And people are trying to develop a lot of machine learning approaches here as well, but it's still quite challenging. Signature analysis is also quite challenging in single cells. There's lots of missing data. A lot of signatures are based on bulk data, and so it's still an area I think that needs a lot of development. Identifying common gene programs across tumors is also challenging. There's a lot of tumor heterogeneity just patient to patient, so kind of cutting through the data. People are starting to use NMFs a lot and other methods, but that can be quite challenging. Integrating different data modalities. If you don't run an experiment where you actually measure it from the same cell, you can also try to computationally integrate things later on, but it has a lot of challenges. Inferring DNA copy number is still challenging. I described one method to do so, but also an area that needs more development. And then reconstructing trajectories, still a big problem in the field. And then one last thing on the kind of discussion of data science and single-cell genomics. Another big project that you might be aware of is the Human Cell Atlas. This is a kind of international project that's mostly grassroots. There's a few people kind of leading it at the Broad and Sanger and other places. But this is really like the human